So picking up where we left off, the electron transport chain, I believe we were here. We just used it as an overview, this slide. So now we're ready to move into the actual process. So we'll look at the structure of it first. And as you can see in the diagram, we're along the inner mitochondrial membrane. So it is a phospholipid bilayer because it is a membrane. And then there are proteins embedded within it. These proteins are acting as electron acceptors. They're going to gain electrons. They're arranged in the order of increasing electronegativity. So the weakest attractor of the electrons is positioned first, and its name is NADH dehydrogenase. It's at the start of the chain. And yes, you do need to know the names of these proteins that make up the electron transport chain. So it's at the start, the NADH dehydrogenase, and the one at the end is cytochrome oxidase. And because of the folding of that mitochondrial membrane with all the cristae, you can get multiple copies of electron transport chain across the membrane. It's not like every mitochondria just has one electron transport chain. It would have hundreds or thousands of them. And that is because of that increased surface area with the folding of the inner mitochondrial membrane. So I'll use the text here as an overview, and then we'll start to look at quite a few diagrams to go over it. So the NADH and the FADH2, they're going to drop off their electrons to the proteins in the inner mitochondrial membrane. So they're going to get reduced. They're going to go back to being NAD plus and FAD. The weakest electron attractor is at the start, the strongest at the end. We saw that in the previous slide. Each component, each protein in the electron transport chain, when it gains an electron, which initially came from the NADH and the FADH2, it becomes reduced. But then it passes the electron to the next protein in the chain, and then it becomes oxidized. At the very end of the chain, there's a molecule of oxygen, which is very electronegative. And that is going to really attract all the electrons through the proteins. So it oxidizes the last electron transport chain component. When those electrons reach the last component, the cytochrome oxidase, the electrons then get passed to oxygen. So really oxygen is oxidizing the last component and is itself becoming reduced. The energy released moves protons, H plus ions or H plus atoms, which are really just protons, across the mitochondrial membrane. So here's an image showing this process. Here's the intramembrane space. Here's the mitochondrial matrix. And you can see the NADH is dropping off its electrons. The electrons move through this protein. And as they do, a proton is pumped across from the matrix to the intramembrane space. The electrons get passed to ubiquinone, and then they get passed to cytochrome BC1 complex. And as they move through this, another proton is pumped across. Then the electrons get past the cytochrome C, which then get passed to cytochrome oxidase complex, and another proton is pumped across. At the end, these electrons get accepted by the oxygen, and really it's just an oxygen atom that gains the electrons, and therefore the oxygen is reduced, and additional protons that are here in the matrix will get added to that, and you'll get the production of water. So this is where water is produced in cellular respiration. Remember that there is the overall equation of cellular respiration has oxygen water as one of the products. So that is where water is produced. And as these protons get pumped across, and as protons are being used to form water in the matrix, really what's happening is an electrochemical gradient is being created. You can see that there's more protons outside than there would be inside, and that creates this electrochemical gradient. And that gradient will then be used to create the ATP in a process called chemiosmosis. Okay, so we mentioned on the previous slide the idea that NADH gets converted. The NADH that's made in glycolysis gets converted to FADH2. The theory is that because the NADH is in the cytoplasm, it has to get into the matrix. And when it does that, is it passes through a glycerol phosphate shuttle. And in that process, it gets converted to FeDH2. And there's a reason why that's important in terms of tallying up the amount of ATP produced, and we'll get to that. But as you can see, there's the NADH that was made in pyruvate oxidation. There's the NADH made in the Kreb cycle. There's the FeDH2 that was made in the Kreb cycle. These molecules are dropping off their electrons in the electron transport chain, but it's important to note that the NADH, it drops it off at the first protein. So as the electrons from the NADH move through these proteins, 3 protons are pumped across. But the FeDH2 bypasses the first protein and drops them off at this Q protein here. And when it does that, it bypasses the first, so really the electrons that were within FeDH2 only get 2 protons pumped across, not the 3 that NADH does. And that is important because the ratio of protons pumped across to ATP being produced is 1:1. Because every proton that gets pumped across is going to come back through the ATP synthase, and that's going to produce ATP. So because the NADH pumps 3 protons across, it's going to produce 3 ATP, and the FeDH2, because it only pumps 2 across, is only going to produce 2 ATP. So now you have this gradient, right? You have a lot of protons outside, and it has a positive charge. So you have an electrochemical gradient, and that creates this proton motive force that moves the protons across ATP synthase. So they're forced back into the matrix through the ATP synthase. And that process is called chemiosmosis. So there's the ratio I was talking about. Every proton that passes back through the ATP synthase gives enough energy to create 1 ATP. This process is called chemiosmosis. The conditions need to be aerobic. And that means there must be oxygen available because that oxygen acts as the final electron acceptor. It needs to be at the end for these electrons to move through the proteins. And if the electrons don't move through the proteins, then you don't get protons pumped across. If you don't get protons pumped across, you don't get them coming back through the ATP synthase to create ATP. So that's why this is an aerobic process. Oxygen is required. And that's why production of ATP in this way is called oxidative phosphorylation. So here you can see they've just kind of abbreviated the electron transport chain, just shown the first one with the NADH. And all these protons would be pumped across, and here they come back through the ATP synthase, and that's producing ATP, which is in the mitochondrial matrix and would have to get out into the cytoplasm, and it does that by facilitated diffusion. So here's the whole process, showing the NADH and the FeDH2 molecules, dropping off electrons, protons being pumped across, protons coming back through to create ATP. Remember that FeDH2 only gets 2 protons pumped across, so it only would produce 2 ATP, whereas the NADH gets 3 pumped across and would produce 3 ATP. So that brings us to the theoretical yield of ATP. Okay, so you have glycolysis, which produces 2 net ATP. You have pyruvate oxidation, which doesn't produce any. You have the Kreb cycle, which produces 2 ATP. Remember, this is per glucose molecule. So you have a subtotal of 4 ATP by substrate level phosphorylation in glycolysis in the Kreb cycle. You have the NADH that is made in glycolysis. You have the NADH that is made in pyruvate oxidation in the Kreb cycle, and the FeDH2 that's made in the Kreb cycle. There's the numbers. Notice that with glycolysis, NADH, it says 4 or 6. This particular textbook shows 4 or 6 ATP produced. It would be 4 if you go with the theory that the NADH must be converted to FeDH2 when it enters the matrix. It'd be 6 if you went with the theory that it doesn't. So there's competing theories out there. So we're going to go with the idea that it does get converted to FeDH2. So only 4 would be produced. And then you can see the ratio of the NADH is 3:1 for ATP production, and with FeDH2 it's 2:1. That gives you a subtotal of 32 or 34.